Welcome to this session of the Sustainability Speaker Series. I'm thrilled to be here. My name is Valerie Miller, and I'm going to be your moderator today. I completed my PhD in land reclamation at the University of Alberta in 2019, and I'm now the coordinator of the Land Reclamation International Graduate School and the Outreach and Engagement Coordinator for Future Energy Systems. Quite a mouthful. But today we are going to be spending our lunch hour with Stephanie Chute-Ibsen. Stephanie likes to refer to themselves as the scientist that Edmonton built. Stephanie completed their undergraduate at MacEwan University before continuing to the U of A, where we're thrilled to have you, for graduate school. Stephanie is currently working on their PhD in land reclamation and remediation. And, fun fact, loves fun facts. For example, did you know that more than 90% of all living animal species are invertebrates? Yet people know so little about them. Not after today. In addition to exploring soil invertebrates, this talk is going to look at soil, plants, the science behind land reclamation, and sustainability. Our planet is being damaged, and this Earth Doctor is prescribing some creepy crawlers. Before we dive into the fascinating world beneath our feet, we want to start by honoring the land and all the ways that it has given us life and honoring the first peoples of this land. As we think about reclamation and restoration, it is essential that we think beyond the settler science that we have learned and seek ways to restore the spirit to the earth through centering Indigenous voices and Indigenous science. We are grateful to the Indigenous peoples that have been and continue to be stewards of the land. The University of Alberta respectfully acknowledges that we are located on Treaty 6 territory and Métis Nation Zone 4, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to influence our vibrant community. Now, before we start, there are a couple of housekeeping items for us to talk about. If you have any questions during Stephanie's talk, pop them in the chat and we will address as many as possible during our Q&A at the end. Additionally, you will notice that we are recording. That will only be myself, Stephanie's cameras. So as an audience member, you don't have to worry, you're not going to be recorded, but that video will be shared on the Sustainability Council's YouTube channel, which you'll be able to find the link in the chat, so that you can watch this again, because you all know you're going to want to watch this over and over again. So I don't want to take any more time. Stephanie, are you ready to go? Awesome. We can see your slides.
Excellent. And everyone can hear me okay?
Absolutely.
Also excellent. All right. Thank you, Valerie, for that introduction. Yes, I'm sure people will want to go back and just rewatch this video. So very excited and thankful to the Sustainability Council Lecture Series for having me today. And we're going to be dishing the dirt on soil invertebrates and how they relate to sustainability. So just like with all research, you're not going to let me advance my slides. There we go. Just like with all research, it takes many parts, many partnerships, and I like to acknowledge those right off the bat. So the Land Reclamation International Graduate School, as well as Future Energy Systems, and then partnering with Capital Power and the Royal Alberta Museum for this work. So even though I'm the scientist at the screen, there is a lot of moving parts and funding behind the scenes as well. So getting started, because we're talking, this is all about sustainability, a little bit of vocab. So when we talk about environmental sustainability, there are various definitions, and these definitions have evolved a lot over time. So the United Nations was kind of the first one that came up with this definition. And the whole concept behind environmental sustainability is that we want to act in a way that ensures the natural resources that we are using and are available to us today are also going to be available to future generations, so that they can have a similar standard of living or a lifestyle that is equal to the current generations. There's also been the International Union for Conservation of Nature. Their whole idea around environmental sustainability is, how can we improve the quality of human life currently while staying within the carrying capacity of these Earth supporting ecosystems? So this whole idea of how can we use these resources that are available to us, but in a way that is not going to overstress our planet, and in a way that means that these resources will still be available to future generations and we're not using it all up. So a lot of the challenges that we have with reclamation is that the challenges with environmental sustainability, sorry, is that we want to continue to be sustainable, but there are big challenges to meeting this standard and this goal of being sustainable. And just looking at some of these headlines, if we're being sustainable and we're not going beyond our carrying capacity, we shouldn't be seeing headlines like this, but we are. So we know that we are currently in a mass extinction event. We are losing a lot of wildlife and a lot of different types of species. We know that there are challenges that we constantly face with climate change. So a lot of the floods happening in BC, droughts happening. We still have a lot of environmental challenges, and these are going to continue to be hurdles and risks that we need to overcome if we want to achieve environmental sustainability and the goal of environmental sustainability. So we know that we have these challenges, and if we are going to address these challenges, we need something to deal with the fact that we are losing habitat, the fact that we are dealing with water shortages. So how are we supposed to achieve environmental sustainability when we know that the actions that we are currently doing are damaging the environment? So how do we balance those? And one of the things that the UN report on sustainable development goals is one of the answers and one of the strategies that we can use to address some of these issues is restoration or reclamation. So they have decided, here are our problems, here's where we would like to be. We want to be sustainable. And basically one of the major strategies to address this is going to be through land reclamation. Wow. That is interesting, because a lot of people that I talk to, if I said land reclamation or land restoration, would probably not know what I'm talking about. So land reclamation specifically is one of the methods that's being proposed, not only by the UN, but it's one of our methods, one of our tools to try to counter the effects of the human activities and promote sustainability. And what it is it's the process of taking these damaged or degraded lands and returning them back to something that has an equivalent land capability. Restoration is in the same vein as that, in that restoration is taking a damaged or degraded system and returning it to exactly what was there before. So if you had a boreal forest now, if you are going to restore it, that's what you would want to get. Reclamation is not necessarily restoring it back to exactly what it was, but more so something that is a healthy, functioning ecosystem that has a similar land capability. So it really is kind of seen as the way that we can bridge the gap between our natural resource exploration and achieving those goals of sustainability. So in this picture here, this is a picture up in northern Alberta with the oil sands. So a lot of times, if you are familiar with land reclamation, that's what people think of. It's commonly associated with mining and other types of resource extraction. But the important thing is that it's kind of our tool to counteract the negative effects of our human activities. So great, we want to be sustainable. We know we're kind of falling short. So land reclamation and restoration is how we can hopefully achieve being a little bit more sustainable in the long term. So reclamation in Alberta, let's talk about that, because let's be honest, Alberta is the center of the world. It's not Toronto. So when we think about activities or areas that we would need reclamation in Alberta, it's actually probably more than you would think. Yes, we can use it for coal mining. You can see in the picture pipeline laying, well pads, oil extraction. But land reclamation is also useful in urban settings. It's useful with renewable resources like solar panels. So anytime there is damage to an ecosystem or an ecosystem is being degraded, sometimes that's by human activities, sometimes it's by natural activities, land reclamation has a position to hopefully reclaim and build that land back to what it was there before. So there are lots of opportunities for us to engage in reclamation in Alberta. And specifically, what I'll be talking about is engaging with land reclamation and mining, natural resource exploration. So the reclamation that's occurring in Alberta, there's always a question of, are we doing a good job? So if we want to be sustainable, and reclamation is going to bridge the gap between mining and sustainability, it's important that we know that the work that we're doing is effective. And there's always been questions among land reclamation scientists and environmental scientists of, is the work that we're doing in reclamation enough to reach those sustainable goals? Are we able to successfully reclaim this land after it's been damaged? And what's a little bit difficult is that there's a lot of reports, there's studies, there's people's opinions coming out that potentially the reclamation that we're doing and the methods that we're using and the sites that we say are being successfully reclaimed might not actually be meeting the standards that we hoped they were. So there's starting to be a little bit of a concern that the way we are doing reclamation may not be that silver bullet we were hoping for. It may not actually be fully bridging that gap between the natural resource exploration and achieving that all perfect goal of environmental sustainability. So I want to just back up a little bit because I want you to understand the process of land reclamation and the kind of the steps that we go through in order to heal this land or bring back this degraded land. So we start with identifying the disturbance. So it can depend on where we are. You might have a disturbance like a well pad, extracting some SAGD, extracting some oil sands. So we need to know what type of disturbance we have because that helps inform us as to what challenges we're going to face. So if you deal with something like mining where they've dug a very big hole, you will have to deal with that versus a well pad where they put a pad down. As long as we know our disturbance, we can come up with an accurate plan on how to mitigate that disturbance. Once we identify the disturbance, we want to determine what is going to be our end land use. So if we're looking at the picture here before, if you have a well pad in the boreal forest, likely your end land use is going to go back to boreal forest. It would not make any sense to put an amusement park in the middle of the boreal forest or a community garden. So we plan our end land use based on the type of disturbance and the surrounding ecosystem. The next step that can happen is remediation. So it can be potentially, there can be contaminants on site. So there could have been some kind of spill. There could be high salted soils. So if there's any type of contaminant that needs to be remediated, and there's a variety of methods that we can use to kind of get rid of or bring those contaminants down to acceptable levels. And once we've removed any potential contamination, then we do the soil reclamation. So depending if you have a very large pit, you might need to fill that back in. If you have a well pad, you need to remove that well pad and basically build the soil back. So again, depending on the disturbance, that soil reclamation can take various forms. Sometimes you have soil that has been sitting or stockpiled for a long amount of time. It may not be overly healthy, so you might need to add amendments. But again, that's all stuff that we plan, keeping our end land use in mind. And once we have healthy, clean soil, then we go in and we plant our vegetation. So that again is coming up with the plan, depending on our end land use, if you're going to use native species, non-native species, a mixed planting approach. That is the next step. And then the final step, but this is also a step that is going throughout the entire process, is monitoring. So when we are putting our soil, when we are remediating, when we are revegetating, we are constantly monitoring the system to make sure that it is staying on the trajectory that we want, and that all of our thresholds are being met so that the soil is healthy and not impeding vegetation. So this whole idea of monitoring is happening throughout the project, but it becomes very important towards the end of the project, because in order to be deemed successful and receive a reclamation certificate, you have to monitor the site and show that the conditions of your reclamation site are similar and close enough to an offsite reference. So the final step, even though it's happening throughout the process of land reclamation, an important step that finally determines have you been successful or not, is the monitoring. So reclamation monitoring, specifically in Alberta, the current criteria that we monitor and that's required is we look at landscape, a lot of other things, but the primary criteria are soil and vegetation characteristics. Excuse me. We go in, we measure the soil, we make sure that there's, nothing jumping out at us. We go in and we look at the species of vegetation. We make sure there are not too many weeds. And for the most part, as long as those are similar to the offsite reference, then that is considered a successful criteria and you are able to get your reclamation certificate. However, as we start to evolve and we realize that we need sustainable ecosystems and we want ecosystems that are very high in biodiversity, there's concern that the current criteria that we are using may not actually be reflecting an ecosystem's function. It doesn't really give us a lot of information about its sustainability and its resiliency. And that has really changed locally here, but there's been a very large global shift and a global focus in how we reclaim and we restore lands. So there's a bit of concern that perhaps the way we are measuring our indicators may not be giving us a complete picture. And some of those headlines that I showed and studies and articles coming out are suggesting that the sites that we thought had been reclaimed successfully, when we go back, they may not actually be hitting the thresholds that we had anticipated. So that makes us worry a little bit, given that, again, we want to have environmental sustainability. So like I said, reclamation monitoring, it does really focus on going in, looking at what the soil and vegetation is doing. And then as long as those two are hitting your thresholds, our formula says that that should give us a successful reclamation site. But again, if we want to be measuring more specifically the function, the resiliency, and the biodiversity of system, I think that we need to be adding something into that, and something that could give us a more detailed picture of an ecosystem. And because all of you can read the title, it's soil invertebrates. So that is the component that I think is missing in the way that we're monitoring, and is what could potentially bridge that gap to make reclamation the actual silver bullet we're hoping it is to bridge the gap to sustainability. So when I talk about soil invertebrates, what is a soil invertebrate? So it's a beautiful blanket term I love to use because it encompasses everything. So soil invertebrates means they don't have a spine. And I like to make a joke that maybe you've dated someone that is similar to a soil invertebrate and they also didn't have a spine. But meaning they don't have a spine, a lot of that means that sometimes you'll see things like earthworms or nematodes, and some of them don't have a spine or a skeleton, but they make up for it by having their skeleton or their spine on the outside. So invertebrates also incorporate things like insects. So insects have a really hard chitinous outer shell, and that's what acts as their skeleton instead of an actual spine. So we have ants, beetles, all of those insects. So insects, anything with 6 legs. But they also include such things as springtails. So springtails are a hexapod, so they also have 6 legs, but they are not an insect. It includes such things as mites and spiders. So they also have their skeleton on the outside, but they have 8 legs and they're in a completely different group. But a lot of these interesting little friends live in our soil. And what makes them incredibly unique and special is that all of these groups play very different roles in an ecosystem. So in a group of soil invertebrates, you can find predators, parasites, herbivores, pollinators, macrophages. So they fill various roles in an ecosystem. And that makes them not just ecologically important, but also economically important, right? When we think about pollinators and stuff like that, there's a high economic value to these soil invertebrates. And what's also great about them is, in addition to, them providing ecosystem services, they also have very diverse body sizes. So I think you can see, even though they're all invertebrates, they all look incredibly different. So they have different body sizes, they have different growth rates, they have different generation times. And for the most part, their population sizes are very large. They have really high reproductive potential. Their generation times are really short, and they can occupy various positions in the food web. So just taking a scoop of soil, you can have various different invertebrates with different positions in the food web, different generation times. It is incredibly, incredibly diverse in so many different ways. So more specifically, when we look just in the soil, there's 3 main components of what make up the soil invertebrates. So we have the microfauna, so really, really small. If any of you know water bears or tardigrades, they're the ones that, they're adorable little microscopic individuals. They can survive in space. Those are in the microfauna-ish, bacteria. So really, really small individuals. And then on the opposite spectrum, we have macrofauna, so quite large, things like centipedes, earthworms, slugs, snails. So those are on the macrofauna. But we see that a majority of the soil invertebrates fall into the middle category. They're the most abundant. So that's the mesofauna, and that includes things like insects. It includes acari, which are soil mites, and it includes things like the columbolans, which are springtails. And what's interesting is I say soil invertebrates, and you look at all of these different groups. And what this image and what is hard to understand is that each of these groups really represent such a specific area. So insects, even though it's a group, we can go further down and look at just beetles or just ants, and then we can look at specific species of beetles. So each of these groups represent a really diverse group where you have specialized scientists in each one of these groups. And what's also interesting is that some of these groups, like insects, are very well understood. We have a lot of resources, we have a lot of research about them, we have a lot of resources available to help identify them. So beetles is one example. We know a lot about beetles and they're very well studied. But on the opposite side of the spectrum, if we go to soil mites, they are not very well understood. In Canada right now, there's about 55,000-ish species of soil mites that have been described, but a conservative estimate is that the number of mites that have not been described, so that means, identified and figured out what species they are, is actually close to 1 million. So there's a huge disparity and a lot of knowledge that's required for each one of these little individual groups. And it can be difficult to pick what group to focus on, because I like to make the joke that if you want to know about these groups, you have to be Liam Neeson from Taken. You have to have a very specific set of skills in order to understand these. And so each of these groups represents very specialized scientists. So the soil biological community is critical to soil quality. It's essential. So the soil invertebrates, like I said before, they're ecosystem engineers. So they help with decomposition. They interact with the microbes, as we can see in this image here. They help form the soil, they help modify the soil, they make the soil structure better, so that's better for plants and all other living things. They help with nutrient cycling. They assist in a lot of the cycling of biogeochemicals. They help with carbon and nitrogen fixing and sequestration. They can help with water purification and storage. So they are involved in every aspect of the soil and almost every aspect of these ecosystem processes. And so it becomes so incredibly important because their diversity and composition has been shown, because they're so involved, that it's directly linked to the ecosystem's health, its function and stability. And then research has shown that, the quicker we have the abundance and the diversity of the soil biota, the quicker we can get that established in our sites, then the ecosystem recovery timeline will be significantly shortened. So we know that they are incredibly diverse and involved in every aspect of these ecosystem processes and these soil processes. So as indicators, we always want to know what makes a good indicator, because we want to be able to go in, measure only a few things from an ecosystem and have it give us a complete picture. So using soil invertebrates as indicators is not really a new concept. It was used in aquatic systems. It was used to help assess, the health of freshwater. So it was really common in aquatic systems, and now it's slowly starting to creep onto land and being used to monitor changes specifically in agriculture. And these, using soil invertebrates as indicators, there is research on using them in response to mining and human activities. That's being used in Australia. They've been using it a little bit of the research. It's happening with brown coal mining in Germany, and it's happening a little bit in forest restoration in China as well. And to be a good indicator, we want these indicators to be sensitive. So we need them to respond quickly to problems in an ecosystem if possible. We want them to be present over a wide geographical area, and we need them to be continually accessible. So regardless of the time we come in, we should be able to take a sample and find our indicator. And the good news is that soil invertebrates really do meet the major criteria of what makes a good indicator. So they're really sensitive to management practices, fertilizer application, pesticides. If you change the vegetation, they are going to respond, and they respond much quicker because of their short generation times, because of their diversity. They respond quicker than vertebrates, than most amphibians, than plants. So we know that they are really, really strong indicators. And because they are so sensitive, they can almost act as an early warning system. So if there is something unhappy in your soil, if there's a contaminant level or a pH in your soil that's not going to be beneficial, the soil invertebrates will respond much quicker than, let's say, the plants or the soil that we're measuring in itself. So the presence or absence of these specific groups can greatly affect the outcome of a reclamation project. So that being said, we know that they are amazing indicators, but there still are a lot of hurdles to overcome to include them. The fact that there's so many and we don't know which one to focus on. So we had our research location nearby, and we wanted to look at and compare what is happening in reclamation sites versus a reference site. So we did our research out at the Genesee coal mine. It's just southwest of Edmonton. And they currently have 2 active pits, and the coal mine from there is contributed to the Genesee generating station, which actually helps supply a large portion of electricity here for Edmonton. And prior to this disturbance, the area there was mainly, a lot of land was used for agriculture. But some of the areas were also fragmented forests and peatlands. And a large component of the reclamation that is occurring out there is focused on agriculture, but Sherritt Coal has made a commitment to try to reclaim more of their disturbed lands back to a forested landscape. So we decided to jump on that opportunity and look at some of their research sites. So we had 2 reclamation sites that we looked at. So if you look at this blurry map, we have Highway 770 that kind of goes through, that's the line that kind of transects this image. And we have on the one side of the highway a site for spruce reclamation. So they placed soils there in 2010, and then they came and planted spruce trees in 2013. And then we have on the other side of the highway an aspen reclamation site. So they placed soils in 2009. They added a lot of amendments. So they added straw and coarse woody material, as well as some direct forest soil. And they didn't actually do any planting. They just, because they had direct soil placement, they allowed the vegetation to kind of come back naturally. And then we have our reference site, because again, anytime we're doing reclamation monitoring, we always compare our reclamation sites to a reference site of what we are hoping this reclamation site will eventually become. So the big questions, and the ones I'll focus on for this talk, because trust me, there's more questions than this, is when we looked at that big group of soil invertebrates, right, the mesofauna that make up so much, which of these groups should actually be considered in reclamation monitoring? So we know that soil invertebrates are great and we should include them. They're very helpful. But out of all of those groups, which one should we focus on? And if we can figure out which one is the most important one, or which one meets our criteria, what is the sampling protocol and timing? So if I want to go to industry and say, listen, you need to start including soil invertebrates so that we can be more environmentally sustainable, I have to be able to tell them what group they should focus on, how they're going to sample it, and is there a time when they should sample? So I have to be able to give them a little toolbox so that we can easily integrate it. So the sampling methods, and I just want to focus on the invertebrates because they're the most fun. But we would go into our reclamation and our reference sites. We did all the vegetation assessments. We did all the soil assessments. And then we sampled the invertebrates. And to try to capture as many of the soil invertebrates as possible, we used kind of 3 different methods. So sampling the above ground soil invertebrates, we did that through pitfall trapping. So if you look at the images below, a pitfall trap is a deli container that I would purchase in bulk. You put a little hole in your site, you make sure the container is flush with the surface. You can do this in your backyard. I do it with kids all the time. We use a styrofoam plate and some skewers to act as a roof to keep the water and some of the curious critters away. And we fill the traps with a kill fluid and we leave them open for a week. And because the roof has a little bit of gap between that and the soil surface, what happens is anything that's walking along, a beetle is walking along, it falls into the trap and is killed by the kill fluid. So we would leave those open for a week and then we would collect them. And as you can see from the image, you basically just get beetle or invertebrate soup. We also wanted to capture what are the things potentially living in the litter layer on the soil. So we would also collect a set area of the litter sample and then extract that. And to capture what was the below ground or the invertebrates living in the soil, we would take soil cores to 10 centimeters depth. And the litter and the soil samples are live samples. So we transport them in coolers, we keep the invertebrates alive, and then we put them on an extraction method called a Tullgren funnel extraction, which is the image below. And that is a funnel with some chicken wire. We put the sample in cheesecloth and then add a small container with ethanol on the bottom. And then I flick the lights on and I leave for 4 days. And what happens is those lights create a temperature and moisture gradient. So the sample starts to dry out. And because the invertebrates are alive, I always feel so morbid saying this, as that moisture and temperature gradient starts to happen, they move down to avoid desiccation, avoid dying. And as that happens, they fall through the funnel and get collected in the vial below. So we were able to I killed millions of insects is basically what I'm saying, but it's in the name of science. So we took all of these groups and looked at all of these groups and tried to figure out which one could be the best indicator. So the groups I want to focus on are the groups that we would get from our tall grain extraction. So that's using the live samples, because the pitfall trap groups, while interesting, we did not find major differences between the sites, but here is where the results get interesting. And here's where you can learn a little bit. So the first picture that we see here on the left is an aurabatid mite. So spoiler, 4 out of 6 of these are mites. And they actually make up a large portion of the mesofauna. So it's not surprising that they're the ones we want to focus on. But aurabatid mites, I like to refer to them as little tanks. So they are, for the most part, most of them are highly sclerotized. So their outer skeleton is really hard, and they are like a tank in that they are heavily armored. But like a tank, because they have such heavy armor, they are also slow. So if you are walking around with all this extra weight and all this extra protection, it makes it more difficult for you to move quickly. So they can't move as quickly as some of the other groups that we're going to talk about. And what's interesting is, compared to the other groups, they actually don't produce as much offspring. They don't have, the same life strategy that some of these other soil invertebrates do. So we refer to them as being more K-selected. So they have fewer offsprings, they have longer generation times. So they're more, if I had to say, more closer to humans, even though they're not, versus these other invertebrate groups are more R-selected. So they have quick generation times, produce a lot of offspring. So aurabatid mites are a very interesting anomaly. The next group that you see here in the middle is a group called prostigmatid mites. So if you've ever seen a red velvet mite, that's actually a prostigmatid mite. But a lot of the groups that we found in our sites were these more prey animals. So compared to the aurabatid, you can see it's basically just a gray outline. So they are white, soft-bodied, fleshy, I'm sure very tasty to all the predators. But we found a lot of these smaller prostigmatid mites, more of these prey species. This other group here is called astigmatid mites. So what's interesting about them is a large portion of these are actually parasitic. So they have a phoretic association with a lot of other invertebrates. So they will clamp onto beetles. They have specialized anal and genital plates that allow them to attach. So they have pretty good dispersal, because if you are tiny and can't move that fast, if you just hitchhike off someone, it's much easier to get around. But interestingly enough, we didn't find a lot of consistent astigmatid mites. The one down here below, a little bit bigger, these are mesostigmatid mites. So these very commonly, in the ones we found, were the predators. So they have modified mouth parts that are called chelicerae. So they're very similar to crabs almost. So they have these little pinchers, and it's so that they can grab those delicious prostigmatid mites or, a collembolan, and kind of rip them apart. So those are our predators. So again, just interesting to see that in these just groups of mites, we have prey, we have predators, we have little tanks, and we have parasites. And then these last 2 pictures here are actually from the same grouping. They're called collembolans. So collembolans they have 6 legs, and some of them have a special appendage on the bottom called a furcula. And that's why collembolans also commonly are known as springtails. So you can see it in this picture with the purple individual. It's a little projection, and they curl it in and they use it to flick themselves. So they can actually travel quite large distances, and they can be predatory depending, and we can have really large, big collembolans, but we can also have another family group where they're really, really small and almost similar to our prostigmatid mites in that they're soft-bodied, very fleshy, and an excellent prey source. So these were the major groupings that we found. Why does that matter, and why that we care? Why should we care? So what I just want to show with this graph is that we went in and we took our samples and we wanted to see, okay, what's showing up in these samples? What's the proportion? What are the major groups? Because again, if we want to use an indicator, it needs to be available, consistently available, regardless of when we sample. So we sampled in 2018, 2019, every month from May to October. And interestingly enough, when we look at it, like I said, the astigmatid mites in orange, they're not the ones that are kind of showing up consistently. In some months, they are present, like in May of 2018, and then June or May of 2019, they're barely there. So we didn't find a lot of astigmatid mites in our samples. In terms of the other category, those were things that showed up. They were very sporadic or infrequent. So that was cool things like a spider here and there, an ant, a pseudoscorpion in our forest, which were very cool, but not enough to kind of put them in their own category. But we do see that, you know, the 4-ish main groups, mesostigmatids a little bit, but the 3 main groups that we're seeing consistently and that make up a large portion of our soil invertebrate populations are the 3, right? So we see yellow as the prostigmatid mites, blue being the orobatid mites, the little tanks, and then green being our collembolans, so our springtails. So that was interesting to find that those are the 3 main groups that make up our samples when we went in and sampled them. But that only means so much. So when we were looking at an indicator, it's important that it's different from our reference site. So we have our aspen reclamation site, we have our spruce reclamation site, but what we're looking for is, and we came up with these criteria beforehand, is in order for an indicator or an invertebrate group to be a good indicator, we needed it to be consistent. So consistency in a couple of ways. We needed it to be consistently different than our reference site, because if you're using it to determine, okay, we want to get to the reference site, we need to find one that's always consistently different from the reference site. So that's something that we can work towards. We needed to find a group that was not going to be heavily influenced by climatic events. So if the group of soil invertebrates fluctuates heavily because of a rain event or a change in temperature, that's not going to be reliable. We also needed it to be consistent from month to month. Monitoring for reclamation can happen anywhere, depending on the weather, from May to who knows, right? November even. So we need a group that will always give us a consistent answer, because if you go and you sample in May and you're getting, you know, an abundance of 2,000, and you go and a different group is sampling in October and you're getting 5, that's not consistent. We need something that will consistently tell us the same story, regardless of the month, the time, and regardless of what's happening in the environment kind of around it. So looking at this info is where we wanted to look at what is the abundance of these main groups in comparison to our reference. So where are they similar? Where are they different? So looking at the graph, we can see that the first one I'll draw your attention to is the X, so the mesostigmatid mites. So when you're looking at this, the closer the icon is to 0, that means it's very similar to the reference site. And the further away we get, so positive values indicate that the reclamation sites actually had a higher invertebrate abundance than our reference, and values that are close to 0 indicate that the mean abundance was very similar to the reference. So we know that these reclamation sites are not a forest yet, so we want to find the indicator that's most sensitive to that difference. So when looking at this graph, we see that the mesostigmatid mites are very similar to our reference forest. And that was interesting because those are our predators. And I hypothesized and expected that, you know, they would not be as abundant in our reference site as our old established forest site, because in my opinion, you need the prey to be there and the predators kind of come after. But that's not what we found, that the predators and the mesostigmatid mites were very similar in our reference compared to our reclamation sites. The next group I want to look at is the prostigmatid mites. So that is the blue diamond shape. And that was one of the groups that we saw made up a large portion of the overall relative abundance of our groups. But we can see, just looking at the way that the symbols fluctuate, fluctuate is that prostigmatid mites are not consistent. So we can see that in a couple of situations, in some months, they're very different from the reference site. So in August and September, we're seeing that there's a big difference. But then once we go to October or May, we're seeing that it's approaching that zero line. So even though the prostigmatid mites make up a really large proportion of the overall invertebrate community, they don't have that consistency and that significant difference from our reference site to be a reliable indicator. So the last two to draw your attention to that kind of offer the most promise, so would be the collembolans. Those are the springtails. So the green circle, there is some promise there, but if we look, there are some months where there is not consistency. So in 2019, we see that the collembolans were very, very different compared to our reference site, especially starting from July onwards. However, when we look at 2018, there's some months where it's not consistently different. The spruce site in 2018 actually had a higher abundance of the collembolans. So while they may be a little bit more reliable, it's something that it's not hitting all of the indicator assessments that we have set out. But orobatid mites is where it's at. So looking at those green triangles, we see that out of all the invertebrate groups, it's the one that appears most consistent. So for the most part, we're always kind of sticking around that -40 mark. At no point do the orobatid mites creep up to the zero line, which would indicate that they're similar to the reference site. So out of all of the invertebrate groups that we could focus on, it's showing us here that orobatid mites are hitting the assessment criteria in that they are the ones that actually show up as being different in a reference compared to our reclamation sites. So what does all this mean? Why do we care? Based on our assessment criteria, so looking for consistency, looking for low variability, and looking for consistency in regards to climate changes or precipitation events, orobatid mites right now are our strongest candidate for a soil invertebrate indicator in land reclamation success. And that is with a second runner-up for the collembolans. So they also show some potential as the groups out of all of the groups in the mesofauna that we should be focusing on in land reclamation. What this means too is that there's next steps that are included. So the next steps include not just looking at the invertebrates in isolation, but using the soil and the vegetation knowledge that we have and doing an in-depth indicator analysis to show if the invertebrates are actually a stronger indicator when we compare it to our existing criteria. And then hopefully, once we can show that, yes, there is value and they are a better indicator, creating a toolbox for industry or consultants to implement so that they can start including invertebrates in some of their monitoring. And that would include letting them know how to sample, ways to extract, how they can count their invertebrates, and all the tools basically needed to start including them in our monitoring. So this take-home message, if maybe you zoned out for a little bit because you're like, oh my goodness, I don't care about mites, but you want to share with your friends what you learned or something that you want to take back home, is that some people say space is, the final frontier and unexplored. I disagree, and I think that the biggest area we need to explore and the final frontier is soil. So there is an unexplored and fascinating world just under our feet, probably close to 1 million species of mites that we don't even know exist. So there's so much room for growth and knowledge. The other take-home message is that we know that our planet is being degraded and damaged. And if reclamation is supposed to be the answer and it's supposed to be the silver bullet to help us bridge the gap between where we're at and achieving environmental sustainability, we really need to ensure that our reclamation is successful and that we are setting up these systems for success in the long run, especially because we know that climate change is going to start throwing a whole bunch of curveballs at us in the future. And the last thing is that this research really is just the first step in trying to explore the use of soil invertebrates as indicators. There's a lot of hurdles to overcome and to make a case for including them, and a lot of knowledge gaps that still exist not just in what the species are, but how they respond to a lot of things that we do in the environment. So it's step number one, and hopefully this research will lead to more interest and more funding for future projects in more ways that we can include soil invertebrates. So thank you. That is all I had to say, and we have a little bit of time left for questions.
Amazing. That was a wonderful presentation. I can't believe you don't think we're all so interested in mites. Like, don't we all want to know those millions of mites now? Absolutely.
That's going to be my life's work.
That I cannot wait for the Stephanie Chute-Ibsen species of mite to be discovered.
Actually, it's considered bad form to name it after yourself, so I will not.
Well, you can name it after me then. There you go. So we have lots of questions, and we do want to fit in as many as possible. So we're going to go rapid fire questions.
Yes.
We talked a lot about the challenges of identification, how to actually make it usable. Can we use genetics rather than visual ID for this type of work?
Oh my goodness, yes, I wish we could, because with soil bacteria, I can go in, I can extract a sample, I send it off, they analyze it, and they tell me what is there, what species of bacteria. Unfortunately, it's work that is ongoing, we're trying to do, but a lot of times if you do that, because we haven't even discovered the species yet, so a lot of it comes back as unknown. There's a lot of work coming out of Ontario and some in the States where we're trying to build that database, because ultimately, yes, that would be the best way. Is you don't have to count individual mites, you can just extract some DNA, but unfortunately you have to do the difficult taxonomic identification and discovering them before we can have an accurate database that will give us accurate DNA information. So that is the dream, but it is another major hurdle that we have to do the dirty work of taxonomic identification in order to bridge that gap to get genomic identification.
Like you said, we're at the beginning steps of this. Okay, do you have plans to expand your study to include different types of disturbances and more reference sites to determine if indicators are sensitive to other activities and disturbances and applicable at a regional scale? Is it just your site only that these results are relevant to, or can they be expanded?
Yeah, so that's a great question. Initially, when we had taken on this project, our goal was to include multiple disturbances. So we were going to do coal mining, oil sands mining, diamond mining, and potentially pipeline. But then once we realized how unrealistic that is, so yes, there is more research needed, because different ecosystems, different climatic conditions, this is where it would need to be expanded, right? So, and this is where it opens the doors. So is the reclamation methods too, does that have an influence? Is reclaiming a pipeline, will you see similar invertebrate groups as to reclaiming a coal mine, even if you're trying to create the same ecosystem? So this is where hopefully this starts the conversation and starts to roll out more projects throughout Alberta that encompass different ecosystems, but also different disturbances. So that is hopefully the goal. But right now, yeah, you can't just say, oh, I found this in this area, so it applies widely. That's, yeah. So eventually, yes.
We're not joking about your life's work. I feel like there are enough projects to last a few careers here. Yeah. Next question. Stephanie wanted to know, other Stephanie, hello, other Stephanie in the crowd, wanted to know a bit more about your reference site for your data. If you can share a little bit more about what that reference site was.
Yeah, so the reference site was an undisturbed mixed wood, like aspen fragment, like aspen parkland kind. So it was a nice part of forest that just hadn't been disturbed. Not entirely sure, like it had a beautiful gray luvasol. So it just, and it was close nearby. So that was the other thing, is you want to use a reference site, but you can't have reclamation sites and then a kilometer that way there's a reference site. So we were limited in what was available at Genesee, but this was larger than the reclamation site. So a nice big tract of just, yeah, undisturbed mixed wood boreal. If you want more information, you could email and I could share, but yeah.
Sounds good. And then this question, you might not be able to answer right now, but people wanted to know, when you are looking at invertebrate responses to reclamation, is it Are the invertebrates more impacted by your reclamation activities or by the initial disturbance? Or do we even know that yet?
Oh my gosh, that is such a good question. And I wish we knew. So that's the difficult thing as well, is with reclamation, you have to do a pre-disturbance assessment, right? So someone goes in, looks at the soils, checks for rare plants, we get the disturbance, you do your reclamation and you start. And what we need is research that does look at what is the invertebrate population before disturbance, measure that, do the disturbance, and then measure basically from time zero of when the shovel gets put in. So that would be, if I'm thinking of the sequential flow of things, first we have to show that invertebrates are useful as an indicator and should be considered. And then once that door is kind of opened and they're considering it, that would be the next thing, is how do we narrow down and more information on, are our reclamation practices helping or hindering? Because that's the other thing. We have amazing research in soil amendments, using things like peat mineral mix, using all these different amendments to help bolster our soil that's been stockpiled or may not be healthy. But we actually don't know. We know that it helps with soil properties, but we actually don't know the effect that it's having on the actual soil invertebrate communities. And potentially, if you're adding peat mineral mix, which is more of a wetland soil, what is that and how is that influencing? So we need information in how does the disturbance influence the invertebrate groups, but then also how do our different reclamation practices influence soil invertebrate groups? So yeah, there are endless questions, and I wish we knew the answers to a lot of these, but hopefully this is stimulating some brains and some new up and coming reclamation scientists to tackle this.
Project 5,000 in the invertebrate questions. Okay, we only have a couple minutes left. So our next question is, you are here talking to the public about your work. You mentioned early on that there's often a gap in people knowing what reclamation is. I'm guessing that you think engagement and education about land reclamation is essential. What are the next steps that we need to do in that?
We need to start talking about it more. So engagement, definitely. I think a lot of people want sustainability. They want to be stewards of this land. They want to care for the environment. And the reality of it is that our natural resource exploration is not going away. And even if it did, if we transition completely to renewables, there's still need for land reclamation. So if it's our silver bullet, and I really do feel like it's the way that we heal the planet from our activities, we have to do it well, and we have to educate people about it. So as much as possible, if anyone gives me the opportunity, I try to tell them about reclamation and the importance, and then being involved, right? So getting involved with groups that promote that, getting involved with your local government, and holding those accountable as being proper stewards. So, are we doing enough, and kind of pushing for that? So I push for engagement, but also just for involvement. And there's amazing resources out there to learn. I know Land Reclamation International Graduate School, LRIGS, there's so much content out there. So yeah, engagement is critical, learning is critical, and then pushing our government and pushing the powers that be to really do this properly, have this be the answer that we need it to be to, help this planet.
Absolutely. And you really wrapped up the last question I was going to ask you, like perfectly in a bow in that answer. So last half of a question, you've talked about how individuals can contribute to land reclamation and getting involved. How can individuals learn more about soil invertebrates in their own community? We've got some fans in the crowd that want to know more. How can they do that?
First thing I would do is, why not put a pitfall trap in your backyard? A lot of things that you find there are readily available. The identification and publicly available taxonomic keys for beetles, insects, all of that. There's really cool resources available through soil ecology. So even if you just Google mites, there are textbooks that may be dry, but getting involved and actually figuring out what's below your feet, because soil is the foundation of life. It is critical to our continued survival and health of this planet. So stop treating our soil like dirt, and take every opportunity that you have to learn And if you're interested in soil mites, there's a select group of weirdos. If you Google soil mites, there's a few of us in Canada. And I promise you that if you're interested, they will listen and they will take you under their wing and teach you the weird ways of soil and soil invertebrates for sure.